are now going to start a big area called bioenergetics, or specifically biochemical energy production. We started off with carbohydrate catabolism. We went through glycolysis. We ended up with 2 ATPs from one glucose molecule. And then we took the pyruvates that were produced and went in and through pyruvate dehydrogenase, did a decarboxylation, removed carbon dioxide, and did a redox reaction and put the acetyl group onto a coenzyme A and produced an NADH with each acetyl. So we produced 2 NADHs in that transition reaction between glycolysis and the Krebs cycle. Right now, everything is going to go on in the matrix of the mitochondria. The matrix of the mitochondria, so think of the mitochondria as this organelle with a double membrane. Okay, the matrix is right in the middle here, matrix. Okay, there's an outer membrane, there's an inner membrane, and all of the reactions of the Krebs cycle, we can call it the Krebs cycle, or we can call it the citric acid cycle, or sometimes they call it the TCA cycle, at least in the olden days when I was a student. TCA standing for tricarboxylic acid, because the first product in the citric acid cycle is citric acid, and it has 3 carboxyl groups. So that's why in the olden days, they used to call it the tricarboxylic acid cycle. Krebs is the guy that discovered it. But anyway, these are 3 names of the same cycle. The enzymes involved in the Krebs cycle are right here in the matrix of the mitochondria. For those of you who've had a bio class, you probably remember that the mitochondria is like the energy powerhouse of the cell. This is where the majority of the ATP is produced. Remember how we said for every 1 glucose, you're going to get 30 ATP? Well, so far we've only produced 2, thanks to glycolysis. So that means the rest, all the rest, are all going to be produced here in the matrix of the mitochondria. And you cannot do any of this extra bioenergetics, get all this extra ATP, unless you have enough oxygen. So all these reactions, Krebs cycle, electron transport chain, all of that, everything has to be aerobic. You have to have an oxygen sitting at right at the end of the electron transport chain in order for all these reactions to work. Now, the electron transport chain, which is also part of this chapter, these are 4 big complexes embedded into the inner membrane of the mitochondria. And we'll get into that probably in another video. But right now, let's focus on the citric acid cycle. Now, remember where we left off. Pyruvate in an aerobic environment in human muscle cell gets converted to carbon dioxide, NADH, and an acetyl-CoA. So coenzyme A, which is derived from pantothenic acid, remember it's a B vitamin, is carrying the acetyl group into the Krebs cycle. Okay? Now, what will help you guys a lot is if you print out the Krebs cycle from your book, okay? Or just Google it and print it out. I don't even know if this is clear at all. But, you know, follow along with that because it'll be easier. You're not going to have to know the actual structures of the intermediates, but you should know the names of the intermediates, and you should know what's happening in each reaction. And usually also some questions could come from, is this a 4-carbon molecule? Is it a 6-carbon? Is it 2-carbon? Is it 3-carbon? So know how many carbons are involved, but the actual details of the structure, it's okay if you don't want to know those. Again, full semester biochemistry class, absolutely, yeah, you would have to do all of that. All right, so basically the Krebs cycle or the citric acid cycle starts right here, where this acetyl-CoA is coming right into the cycle. And the first reaction, there's an oxaloacetate, which is 4 carbons, and then the acetyl-CoA comes in, and that's 2 carbons, and they combine to form citrate. Citrate is the first product, and this is a molecule, an enzyme that catalyzes that is citrate synthetase, and you end up with citrate synthase, and you end up with that citrate molecule. So the citrate molecule is a 6-carbon molecule because oxaloacetate is 4, the acetyl is 2. And so once the coenzyme A drops the acetyl in there, it can go back and pick up another acetyl in that pyruvate dehydrogenase catalyzed step. All right, citrate, like I said, is the first product, and that's why they call it the citric acid cycle, because citrate is that first product. Citrate, which is 6 carbon, is turned into something called isocitrate, and it is also 6 carbon. Okay, so that's the next step. So you can again follow along with the cycle. Do you need to know the names of all the enzymes? No. This one is a aconitase. You don't need to worry about the name. But the next reaction is an important one. So the first reaction is oxaloacetate plus acetyl-CoA to give you citrate. The second reaction is just basically isomerizing citrate a bit, removing water, adding water, and then turning citrate into isocitrate. Third reaction is super important. Third and fourth reactions are very important. Third reaction is a redox, and so is the fourth reaction. They're both redoxes. They are both decarboxylation. What does a decarboxylation mean? It means you're removing a carboxyl group and it's going off as carbon dioxide. So in the third reaction, so first reaction was oxaloacetate plus the acetyl will give you citrate. That's the first reaction. Second reaction, you're isomerizing citrate, turning citrate into isocitrate. Okay. Third reaction, isocitrate is turned into alpha Kg, which is alpha ketoglutarate. In the process, you are removing one of the carbons. So carbon dioxide goes off. Remember how we said that a molecule of glucose through cellular respiration uses 6 molecules of oxygen, produces 6 molecules of carbon dioxide, and 6 of water. Well, we already produced 2 molecules in that transition reaction between glycolysis and the citric acid cycle. That pyruvate dehydrogenase, remember, that turned pyruvate into acetyl-CoA, that turned a 3-carbon molecule into a 2-carbon molecule, and then one of the carbons went off as CO2. And so you did that twice because you had 2 pyruvates, you have 2 acetyls, CoAs, so you have 2 CO2s. So we produced 2 CO2s there. We're going to be going through the citric acid cycle twice. Why? Because we had, remember from that one original glucose, we had 2 pyruvates, and then the 2 pyruvates became 2 acetyl-CoAs. So one is going to go in, and then the next one is going to go in. So I'm just going to focus on going through the cycle once right now, and just know that you are, from the original glucose, you're always going to have to go through it twice. All right, so that one acetyl-CoA hooked up with oxaloacetate to make citrate. So this is 4 carbons, this is 2 carbons, it became a 6-carbon molecule. The citrate got isomerized and became isocitrate. This is also 6 carbons. The isocitrate turned into alpha ketoglutarate. It lost one of the carbons, so now this is a 5 carbon. And in the process, you used an NAD and you turned it into NADH. We always put that H plus there to account for that plus, but the reduced form of NAD is NADH. So this is important because remember, we're going to be counting how many NADHs we produced in the Krebs cycle. We already counted that we produced 2 in glycolysis. We produced 2 in that transition reaction between between glycolysis and the Krebs cycle. So we're going to add them all up at the end and we'll have a nice accounting of everything. But right now, let's go through the motions here. All right, so this reaction is catalyzed by an enzyme called isocitrate dehydrogenase. Remember, dehydrogenases catalyzed redox reactions. They removed hydrogens from isocitrate, put it onto NAD, and it became NADH. So isocitrate became alpha ketoglutarate and a carbon dioxide. The next one is a reaction where alpha ketoglutarate, look at your cycle over here, alpha ketoglutarate is now going to get turned into something called succinyl-CoA. You're going to remove another carbon dioxide. So you have succinyl coenzyme A. So another coenzyme A is going to be used here. It's going to be carrying the succinyl, all right? In the process, you're losing another carbon dioxide. So this CO2 and this CO2, again, if you go through the cycle twice, so this is 2 and this is 2, that's 4. We already lost 4 in that transition between glycolysis and the Krebs cycle. So that's the total of the 6 CO2s. So you're getting rid of a carbon dioxide in the third enzyme with the third enzymatic reaction, and you're also getting rid of another one with the fourth enzymatic reaction. This enzyme is alpha ketoglutarate dehydrogenase. Again, looking at your cycle, you can see that alpha ketoglutarate dehydrogenase would be catalyzing a redox reaction, just like isocitrate dehydrogenase did also. Okay, so we are done with numbers 1, 2, 3, and 4. Since we lost another CO2, now we're at 4 carbons. Okay, so we're not going to be losing any more CO2s. We have 4 more enzymatic reactions in the Krebs cycle, and we'll do those in the next video.